Alright, I am putting this video together because I want to give you guys more information on this last lab that we're doing. My mic is a little hazy, so I'm sorry for the terrible sound quality, but we'll deal with it. Okay, so first and foremost, this last lab is based on the polymerase chain reaction, and this is an outline that gives you information on that, so I want to go over that to make sure we all understand what's happening. PCR is a way to take double-stranded DNA, or any nucleic acid. Really, you can do RNA too. And we want to amplify it. We want to get more. And we're going to go through this cycle, and every time we go through a cycle called thermocycling, thermo because we're changing the temperature each time, we're going to double our DNA. So if we start with one strand of double-stranded DNA, and we run through this cycle, we get 2. And then we go through again, we get 4, and 8, and 16, and 32, and 64, and 128. And we keep doubling every single time, and we get exponential amplification of our starting nucleic acid material, right? So to do this, there's a lot of stuff that can go into the PCR reaction, but the most important things are our template DNA that we start with. We add in primers, and we add in polymerase. And so what we do is we take our double-stranded DNA, the template that we want to amplify, and we add in our primers and our polymerase, and we go through this cycle where we denature, so we heat the cycle up to around 95 or 98 degrees Celsius, and this separates our 2 strands of DNA. Then we slowly cool it down and we anneal, and this allows our primers, which are complementary to this DNA that we care about, this allows our primers to bind that strand of DNA. After we anneal, we heat it back up a little bit to 72°C. This polymerase, which is an enzyme that will take DNA and will make a complementary strand in the 5' to 3' direction, right? Is engineered to work best at 72°C. And so our polymerase hits this primer and it makes a new strand of DNA that is complementary to this strand that we started with, our template. And then this cycle continues. We go back here, we denature, anneal, extend, get more, denature, anneal, extend, and we get more and more and more DNA. This is the root of what we're actually doing in lab this week. So if we look actually through our rubric, or not a rubric, our handout, this is what we would be doing in part 2. In part 1, we would be harvesting DNA from technically your own cells. So you take like a cheek swab and you would isolate that DNA using this kit. If you want to get more information on what's actually happening through here, you can look at this video called Zoom PCR Lab 4. This is on the modules for the lab, for this lab on Canvas. Okay, so Leon made this video. He goes through pretty good detail of that. So I'm not going to talk too much about this, but basically you're isolating DNA. At the end, your DNA is now eluted from the column in the tube that you prepared. So you have DNA that you've isolated. In week 2, what we would do is we would do an amplification of that DNA. So what we're looking at is the sex chromosomes, so this 23rd pair of chromosomes, and we have 2 different genotypes that we could have typically in the population, right? We would have an XX and an XY, right? And so all the chromosomes between are the same, except for when you have these different sexes, we have XX and XY chromosomes. So we can do PCR to see how this would, see what we have in our cells, right? So what we can do is set up a couple reactions where we have different primers, right? So if we have a common primer, this primer will bind both chromosomes. Will bind both the X and the Y chromosome. The X-specific primer will only bind the X chromosome, and the Y-specific primer will only bind the Y chromosome. And so the common primer and the X-specific primer will amplify almost a 500 base pair fragment from the X chromosome, right? So this will bind the X chromosome. Then we have about 500 base pairs, and this X primer will bind again, right? And so when we amplify that, we should get about a 500 base pair fragment amplified. The Y-specific will bind the common and the Y-specific chromosome. So the Y chromosome will bind common and Y-specific, and the gap between those is about 340 base pairs, so we should see a 340 base pair fragment from the Y chromosomes. So, what we would expect to see is if you put all these primers in and you have XX, you should see one pretty strong band at about 500 base pairs. If you have XY, you should see both this almost 500 base pair band and a smaller band around 350 base pairs, right? Because we'd have both X and Y, whereas with an XX sample, you would only see this one band. You probably would see a stronger band because there's two chromosomes to copy from. So, what we would do is go through this reaction with the DNA that we got from week 1, okay? And again, you can see a lot more talk on what we're adding in here and why in here, but we're really just doing this thing that I already talked through, okay? We're going to amplify. The next thing we would do is run it on an agarose gel. So you can see in the handout, you see the different things we do. So here's our setup. Instead of this isn't exactly what we would do, but here's the exact setup that was done. We'd run a gel, and we would look at the gel just like we looked at it in the last lab. Okay, but this lab we're not doing like any math or making linear regressions or anything. We're just kind of eyeballing it and seeing what we have. Okay, so here's the results. Okay, and I want to make this very clear in this video that these results that I'm going to tell you to do are a little different than what you're going to find online. Okay, so this is a little bit of the data as processed. This is as you see it on Canvas. Okay, the Canvas says that we have our ladder here, and then we have a female positive control lane in lane 2, a male positive control lane in lane 3, a male student sample, so this should be XX, or this should be XY, I'm sorry, and then we have a negative control with no DNA in lane 5. These down here in your gel, these are really small, these are like 20 base pairs or so. These are what we call primer dimers. So this is where your primers are binding together, and you see you can get a decent signal here, but they're really small. So these are not your PCR products. This is fairly common to see at the bottom of gels if you have primer. So just for your own awareness, if they're that small, that's not what we're looking for. Okay. Okay. Now here's the issue. Okay. With this data. So let's look at this. A female positive control. This is XX. Right? I should see XX. Let's look through this again to make sure. Female genotype XX, male genotype XY. So if I have XX, what should I see? I should see one band X primer with about 500 base pair fragment. Right? Let's go back over here and look. I see two bands in my XX lane. That's interesting. Let's look at our male positive control. I see one band in my male positive control, right? So, again, if I look here, I should see, so for my male should have two, right? Because I should get a 500 base pair band roughly from this X primer, and I should get the 350 base pair roughly fragment from this XY, because I have both the X and the Y, right? So, basically, my male should have two bands, my female should have one. If I look at this DNA, though, my female has two, my male has one. I think when they ran this gel, the samples got mixed up. And I apologize for this, because as per the usual, this is a complete dumpster fire of data that you have. But I think these bands are messed up. Okay? Also, even worse, if we look at our male our male student sample that technically should be run for this, it's XY, but it didn't work. The reaction didn't show up with anything. And then because look at the negative control, right? Nothing. Okay, so this is kind of a dumpster fire. This is as it is presented in Canvas, if you look at all the Canvas data. Here's what I want you guys to go off of, though. Okay? And I'll send this out in an email. I cut off the rest of the gel. Okay? Here's our sizes. Okay? This is your ladder in lane 1. In lane 2, this is XY. So this would be a male chromosome. Okay? Two bands. One's a little bigger than the other. That makes a little bit of sense. In our XX, we have a female chromosome. Okay? Or in lane 3. And we see just this one strong band. It's stronger than the rest. It makes more sense that this is female than this being male. Okay? So let's assume this is what we're looking at here. Okay? 1 is our ladder. 2 is XY. 3 is XX. Lanes 4, was supposed to be an XY experiment, but for some reason nothing showed up. Lane 5 is negative control. I don't even care if you don't show me that anymore. Just show me these and talk through it. Okay? So let's pretend this is your actual data that you were working with when you do this lab. It'll make more sense. I've already gotten an email or two about this as well. So I'm glad you guys are seeing that things don't add up, but let's keep that in mind. Okay? Ladder, male, female. As far as the test sample, I don't even care. Blow it off. It didn't work. Just talk about how these bands, the sizes, if they make sense, why they make sense, and if they don't. Okay? You can still go through here and go to the rubric, right? And still talk about these discussions, right? Explain how the primers were used, what products were expected, why, do they match your predictions, sources of error, ways to improve the experiment, right? Are there any errors? Well, does this gel look like it actually worked very well? Maybe not, right? These bands are not defined really well at all. These very smiley. It was probably run very quickly and probably could be run longer, right? The agarose maybe didn't set up completely well. That's why we have these weird smears in the gel. So you could talk about some of those things. You can just Google, like, what gives me smeary bands on an agarose gel, right? So you could look at some of that and give some of that information as well. In the handout, you also have these questions that would be good to look through, right? So like what are step 3, you don't even necessarily put these in your report, but it's good to look through these if you want to make sure you understand what's going on. But just go off the rubric here. Talk about these in your discussion. All you need for your results are include an image of your gel with the labeled lanes and approximate the size of the band. I would just put something like this in here. Just blow off lanes 4 and 5 if you're in my section because they didn't work anyway. And like I said, I'm pretty sure these were flipped. This makes much more sense to me because you see 2 clear bands here, and that's only going to be there for an XY. Okay? So, with that being said, this is a full report. And just go off this rubric here. Okay? I'll send this file out to you guys. Fill out this rubric. Just do a quick thing. I'm going to be leaning on this one as well because it just doesn't seem to work very well anyway. Okay? So this is due on the 4th of December, so that is this coming Friday. If you have any questions or issues, feel free to reach out to me, and I'll talk through those with you. Okay? Hopefully this helps, and yeah, let me know if you have any issues.